everyone. Sorry for the delay. Welcome to Histology 101. We had all sorts of technical issues. The computer decided to do automatic updates without asking, and then decided to break after the update. So I had to play around and do a little bit of Googling to find out what was wrong. But we're here, we made it. Welcome along. Thank you very much for your patience. I appreciate everyone being along here. First of all, let me please thank and welcome to our slide box True Blue 5, Histolab, Kimmy, VYD, Histo Geek 1. Thank you very much for the follows. We really appreciate it, and welcome along to our live stream, Histology 101, where today we're going to be looking at the histology of the reproductive tract of people assigned female at birth. So in our last stream, we looked at the histology of the ovary, we looked at folliculogenesis, oogenesis, and all those things in between. And so we're moving on to the reproductive tract today. So we'll probably start with just a very quick broad overview of the reproductive tract. And then have a look at some of the finer details of the histology. So please come along, say g'day, ask your questions. I've got Dr. James Crane here helping out in the chat. So if you see any typing there, that's him helping me out on the side. So maybe we would just do a really quick overview, reminder of what actually makes up the reproductive tract. I'm sure everyone's probably quite well aware of it. You might have noticed that I put a little, this stream is for mature audiences only, because I want to just make sure that we didn't breach the Twitch terms and conditions. And I see a number of streamers who get in trouble. And in fact, when I tried to put the word vagina in the title, it wouldn't let me do that. So apparently we can't mention vaginas. I do think we are allowed to because of educational, but I guess we'll wait and see if we're still on next week. All right, so the reproductive tract is consisting of a number of features. So you have to bear with me because I'm not the best artist. Okay, so please bear with me while I attempt to draw this. As I say, I'm not the best of artists. So I'll just keep it sort of continuous with that. Sorry, I'm not very good at multitasking. Okay, so we of course have the uterine tubes here. So these are also referred to as the oviduct or the fallopian tube, and we imagine we have the ovaries off the side here. They're very far removed. Wow, this is really massive. So we've done quite long. I don't know why I did uterine tubes that long. It looks like Michael Jordan holding the basketball out like so. Anyway, yeah. Oops. Okay, so here's our ovary. Oh, come on, I'm pressing the wrong button, I apologize. So here's our ovary, and then we can sort of subdivide the uterine tube into different regions. Here we have the infundibulum. And the infundibulum contains those little finger-like projections, which we refer to as Fimbriae. So there's those little finger-like projections. They have very ciliated epithelium there. And then we have this region here, which is the ampulla. Ampulla means wide bottle, so it's actually the widest part of the uterine tube. And of course, this is important because this is generally the site for fertilization. So if fertilization generally takes place in this region here. Then we have this narrowing here. This is known as the isthmus. So here's the isthmus. We often, sometimes some textbooks describe another part of the uterine tube here, which is known as the intramural portion of the uterine tube, and that's the part that sort of passes through the wall of the uterus here. So here's the uterus, and we identify different parts. So this is the fundus. So this is the body of the uterus. The uterus also has an isthmus, which is sort of just a narrowing. And then we have, of course, the cervix. Now the cervix has a little endocervix, and coming off the endocervix we have little glands, mucus secreting glands. These are known as the endocervical glands. We'll talk more about those in a little while. So it actually has sort of two openings to it. It has one that sort of faces the uterus, and so this is known as the internal os. And then the opening that sort of faces out into the vagina is known as the external os. And then here we have the vagina. All right, so that's just really sort of the overview of the internal reproductive tract of people assigned female at birth. The vagina, of course, opens up at the vestibule, the vaginal vestibule, and so we're going to talk about all the different components and also how it changes during a normal menstrual cycle. So we're going to start off with the uterine tube, and then we'll make our way down looking at the uterus, looking at the cervix, and finishing off with the vagina. And if time permits, we'll have a look at the mammary, a quick look at the mammary glands as well. And so again, if you have any questions, please pop them in chat. We're very happy to try to answer those questions. All right. So you might remember, if you were here in last week's stream or you caught up with the video that I uploaded to my YouTube channel, Chapman Histology. Don't forget to leave a like and a follow, and don't forget to hit that notification bell. So you might remember this. This was this weird section of the ovary here with this weird bulby thing at the top, which actually happened to be part of the ovarian tissue, but just not in this plane of section. And the reason I'm starting here is because we actually have the start of the uterine tube here. We actually have the infundibulum with the little finger-like projections of the Fimbriae. And you can see a little bit of a cross-section of the uterine tube here. This is very early sort of, end of the infundibulum becoming the ampulla. So if we sort of zoom in here, we can actually see, look how highly folded these are, these finger-like projections. They're basically just an epithelium and a layer of lamina propria here. So you can see they're sort of little floating. These of course are connected, but just in this plane of section they're not. So we sort of zoom in here. There's really not much to see other than this epithelium. This is generally a simple columnar epithelium with cilia. You can see lots and lots of cilia on the surface here. You can see the little basal bodies, which is the sort of origin of the microtubules here that form the core of the cilia. So the infundibulum is a highly ciliated structure. Those cilia create a current that draws the ovulated oocyte from the surface of the ovary and from the pelvic cavity into the uterine tube itself. So you can see them all sort of surrounding the ovary, but you can see they're actually not connected to the ovary. So they're not actually connected. They're just sort of separate, and the oocyte that is ovulated into the pelvic cavity on the surface of the oocyte, and it's drawn in to the uterine tube via these finger-like projections and their cilia. So this is the uterine tube here. So if we move to the next section, here we've got our section of the uterine tube. And as we just talked about, the uterine tube has 3 or 4 regions depending upon which textbook you're reading. once we move from the infundibulum, we see quite a dramatic change in structure of the uterine tube, and we sort of describe it as having 3 layers. So if we have a look here, we can make out 3 layers. So we have a mucosa, we have a muscularis, and then we have a serosa. You can sort of see, you imagine that they sort of have a mesothelial lining. You can see some of the blood vessels here as well. So the mucosa consists of a simple columnar epithelium, ciliated columnar epithelium, and an underlying layer of lamina propria. So this is the lamina propria here, and the epithelium, of course, is on the surface. Now, in the ampulla, the mucosa is extraordinarily folded. It's really, really folded to increase surface area for contact with the ovulated oocyte. If we were actually to see the ovulated oocyte, it would probably be about that size. So it's actually a bit of a maze for sperm who are trying to find the oocyte to try to find it. And interestingly enough, over time, if we sort of look at the evolution of mammals, and this is a really bizarre conversation to have, I suppose, in a histology unit, but the evolution of mammals, we've got sort of evolved from reptiles, which are sort of external ovulators. They created eggs like monotremes, and then the eggs reduced in size, and the marsupials, sort of the intermediary, they have quite a large oocyte. But then the what we call eutherian mammals that have placenta like humans, have a much smaller oocyte. And in order sort of to increase that surface area so that it can actually contact sperm, they have this very well-developed corona radiata and extracellular matrix around the outside of that ovulated oocyte. And then it's thought that within those corona radiata cells, we have lots of chemotactic factors, which are sort of stimulating sperm, which are being housed within the uterine tube to come and find the oocyte within the uterine tube. So if we have a look at this in a little bit more detail, so we can see sort of how folded this is. So here's one fold. So we often talk about the ampulla of the uterine tube having a primary, a secondary, tertiary folds within the nature there. So we sort of zoom in. We can actually, it's not always that easy to identify, but there are generally 2 cell types found within the epithelium of the uterine tube. It's described as a simple ciliated columnar epithelium, although it's often, you can see cells at nuclei multiple levels, so I suppose the pseudostratified isn't out of the question as well. But we generally have 2 cell types. We have the ciliated cells, and then we have the non-ciliated cells, also known as peg cells. So they often appear as if someone's actually sort of pegged them into the surface here. Now, they're not, as I say, not always that easy to see. Unless you've got a nice section. We can certainly see the columnar nature of this epithelium, certainly here, and you can see the cilia here. The peg cells, on the other hand, not always that easy to see. If we were to see a scanning electron micrograph of this, ciliated cells, of course, stand out by having all the cilia. The non-ciliated peg cells are characterized by having short microvilli on their surface. Now, those peg cells are responsible for the secretion of factors which promote fertilization and early embryonic development. We can hear some people talking in the background as they walk past, ignoring the sign that a recording's going on. So within the lamina propria, we have a loose connective tissue, we have capillaries, we can have large vascular vessels here as well. So this is a nice large, it's almost like a lacteal. It's a large blind-ended lymphatic capillary here as well. Then we have our muscularis. It's generally divided into an inner circular outer longitudinal layer, and what happens is as we move along towards the uterus, we get several changes happening within the structure of the uterine tube. Now, I don't have multiple sections of the different regions of the uterine tube, I've just got this one. But what happens is that the folding of the mucosa decreases as you move closer and closer to the uterus, and the thickness of the tunica muscularis increases. So it starts off relatively thin, muscular wall in the ampulla, and then becomes more and more thicker as it becomes closer and closer to the uterus. And of course that makes sense if it's going to eventually become continuous with the uterus, which has a very well developed muscle layer in the form of the myometrium. So that's, again, you can see just how complex this folding is with this uterine tube here. You can see some of the peritoneal covering on the outside and the blood vessels, the vasculature associated with the peritoneal covering. There would normally be a simple squamous lining here. We can just kind of make out a little mesothelial cell there, but they don't always stick around when we process for microscopy, unfortunately. But yeah, that's what we would expect. So I don't suppose anyone's got any questions. I haven't seen anyone actually typing in chat yet, so other than James. All right, okay. So we're going to move on to describing the histology of the uterus. And I think in order to put this into context, because we've actually got 3 phases of the uterine cycle to talk about, it's important that we have an understanding about what actually is happening within the uterus during the normal menstrual cycle. Which particular hormone activate the Fimbriae, and do we need to know them? Tyranny, estrogen is the important hormone for ciliogenesis. So the maintenance of the cilia is provided by the estrogen, secreted by, of course, the developing follicles within the ovary. Estrogen drives and maintains the cilia, and of course the cilia are really important as part of the oocyte or gamete transport. So if we're having a look at this here, we're actually getting 2 sort of things happening. We're getting the cilia current drawing the oocyte from the outside towards the uterus. So should fertilization take place, it takes place here in the ampulla, and we're also getting peristaltic contraction of that muscularis. So we've got these 2 gamete transport forces working. We've got the cilia creating a current, and we've also got peristaltic contractions moving this ovulated oocyte down towards the uterus, so implantation can take place if fertilization had taken place. So when the sperm are coming, they're deposited in the vagina, they make their way through the os, assuming there's the correct conditions, through the external os, through the endocervical canal, into the uterine cavity, along the length here, and then as they migrate through the uterine tube, they're swimming against the current created by the cilia and the peristaltic contractions created by the muscularis. So it's actually a good way for the female reproductive tract to weed out the poor quality sperm, and we know that humans actually do have very poor quality, at least in morphology, poor quality sperm. And because of the mating systems that we have, no selective pressures to actually produce good quality sperm. So, I mean, that was a lot more information than you actually asked, so hopefully, estrogen is the answer to your question there. All right, so we're going to have a look at the structure of the uterus and how it changes during the normal menstrual cycle. So we're going to have a look at that. So I might just quickly save this one in case we need to come back to it. But let me close that. All right, so the uterus is actually divided into 3 layers. We have the endometrium, we have the myometrium, and we have the perimetrium. Now the endometrium can be further subdivided into 2 layers. It's divided into a stratum functionalis and a stratum basale. And then we can actually further subdivide the stratum functionalis into a stratum compactum and I've forgotten what the other one is, but we don't really talk about that in the unit that I teach here. Myometrium is made up of 3 layers of smooth muscle. And then the perimetrium is made up of connective tissue and or a serosa. So depending where the section's been taken from, so if you're talking about the fundus or we're talking about the body of the uterus, most of the time that's going to be a serosa. But then as we move more distally, it's more likely to be a connective tissue which is binding it to the rectum posteriorly and the bladder urethra anteriorly. So we sort of can start to think about the uterus over our 28-day cycle, normal menstrual cycle. And so we sort of divided into 3 phases here. The first of the phases, so the start of the first shedding of the endometrial lining is day 1 of the normal 28-day menstrual cycle, and so this is known as Menses. Then as that regrows, it becomes part of the proliferative phase. And then over here we actually have the secretory phase of the menstrual cycle. So this is the uterine. My head in the way?
Yep.
Yeah. All right. All right. So, yeah. Am we still on screen? No, we're not, are we? All right, let me just adapt that, sorry. All right, so this is the 28-day cycle. We divide the uterine or menstrual cycle. I'm looking at the camera and of course I'm not on screen. We've got menses for the first 5 days. Then we've got the proliferative phase and the secretory phase. So if we sort of think about the endometrium then, we have the sort of the stratum basale, which is sort of generally always present. And then we actually can have the, we might pick a different colour here. So we actually lose the stratum functionalis. Here. So I'm just going to keep drawing these. Okay, so if this here in this sort of lighter pink is the stratum functionalis, and then this other colour down the bottom is the stratum basale. Oh. Okay, I just deleted all that and there's no way to go back. There's no Control Z.
Bruh.
All right, I'm having a good day, ladies and gentlemen. All right, let's have another go. I apologise for that. All right, let's see if we can do this at a much faster speed here. So we had Menses, we had the proliferative, and we had the secretory. Okay, and so we had our stratum basale, and then we had our endometrium, Okay. So here's our stratum functionalis, here's our stratum basale. Now, a really important part of this as well, I almost pressed clear again, goodness gracious. We have our blood vessels, so we have our blood vessels here. And so down here we actually have our straight arterioles. Okay, so we have our straight arterioles. And then from the straight arterioles, now these are damaged because of course we've had vasoconstriction of these straight arterioles. And then branching from the straight arterioles, we get our spiral arterioles. And of course, what's happening during this time is that we're preparing the endometrium should implantation take place. Okay. So as I was saying, we've got our straight arterioles here. And then here in the stratum functionalis, we have our spiral arterioles. Okay, so what happens during this time, and again, we need to actually refer back to our previous menstrual cycle and what's happening with our hormones here and within the ovary. So everything that's sort of driving this uterine cycle here is actually the ovarian cycle and the hormones from the ovarian cycle, which is driving it. So in the previous cycle, I always struggle to find the words to sort of describe this here. So chronologically, we have these three phases of the uterine cycle. We've got the Menses phase, the proliferative phase, the secretory phase. What I'm going to do is actually I'm going to start at the proliferative phase, even though we know Menses is the first chronological phase of the menstrual cycle. So if you don't mind, what's happening here is FSH is starting to rise. Here, FSH from the ovarian cycle captures already developing follicles, secondary follicles, eventually selecting one dominant follicle that then goes on to eventually become ovulated. So as FSH captures those developing follicles, the levels of estrogen begin to rise. And estrogen is what's bringing about this proliferative phase. So this regrowth of this shed stratum functionalis, remember this is the stratum functionalis on this side and the stratum basale down here. So during menses we shed the stratum functionalis right back to the level of the stratum basale. There are glands which sort of, I haven't sort of drawn here, but exist down in the bottom of these. So some of these glands actually extend down. I haven't drawn them sort of deep enough. So these actually extend down into the stratum basale here. Okay. Yeah, so you get the idea anyway. Okay. So the epithelium from the base of these glands actually regrows out onto the surface of the endometrium here. So the endometrium, this stratum functionalis, consists of an epithelium and stroma, and within the stroma we get our glands. That's a G, glands, and of course we've got our blood vessels. So this is all within this endometrium here, the stratum functionalis, the stratum basale. So the epithelium that lines these glands eventually regrows out during the proliferative phase, and this is driven by the estrogen, increasing levels of estrogen as these FSH is capturing these developing follicles. So these increasing levels of estrogen drives the proliferative phase. It causes a regrowth of the epithelium from the base of the glands, from the stratum basale, out to the surface. The stroma regrows. We get a regrowth, resprouting, if you like, of our spiral arterioles from our straight arterioles, which exist within the stratum basale. And the glands grow as well. Now, initially, these glands are really just straight tubular glands. And they're really not secretory at this stage. They're more sort of, if we have a look at their structure, we'll see that they're like mitotic figures indicating that they're actually growing. So as I say, this estrogen levels actually begin to increase. Now, these moderate levels of estrogen actually have a sort of inhibitory effect on FSH release. So those levels actually drop. But it gets to a point where estrogen actually reaches a peak. So we get this sort of peak of estrogen around about day 12. So around about day 12, we get our capture of our dominant follicle. The dominant follicle is pumping out lots and lots of estrogen, and that actually has a positive stimulatory effect on GnRH release, and then FSH, but particularly LH. So on day 13, we actually get this big surge in luteinizing hormone, and then eventually within the ovaries, that brings about ovulation taking place. So histo 1, 2, 3, 4, 5, 6, 7, 8, 9. What do the layers look like after menopause if estrogen is driving proliferation? These layers decrease quite significantly unless the person is actually on hormone replacement therapy. These actually decrease to basically the stratum basale level. So we tend not to have a lot of these glands and so on, just very shallow glands and so on. So it's not certainly not going through the sort of normal changes that we see that are driven by those estrogen progesterone levels. Hopefully that answers your question there. So we get our surge of luteinizing hormone on day 13. On day 14, we get ovulation, and that remnant follicle within the ovary transforms into a corpus luteum. And so what the corpus luteum is pumping out is a lot of progesterone and estrogen again, but mainly it's progesterone. So what the progesterone does is it drives the secretory phase of the menstrual cycle. So it causes the regrowth, the continual growth of the endometrium, the stratum functionalis. It causes this increased complexity in the structure of the glands. They actually take on what we describe as a sawtooth type of appearance. Oops. That'd be helpful if I could spell. Sawtooth appearance of our glands, and of course they also start to secrete material as well. So we actually see material in the lumen of these glands for the first time, whereas previously we just see these simple straight tubular glands. In the secretory uterus, we see this increased complexity in the structure of the glands taking on this sort of sawtooth appearance, and we see material within the lumen of these glands as well. We get this continual vascularization of the stratum functionalis with the spiral arterioles in the stroma here. And then, so this is, as I say, it's all being driven by the corpus luteum and the progesterone predominantly and the estrogen from the corpus luteum. Now, if fertilization doesn't take place, and normally fertilization happens within 24 hours of ovulation, so around about day 15, and then it takes another 6 or so days, 6 to 8 days for the blastocyst to develop. So the embryo goes through development, becomes a blastocyst, and then reaches the uterine cavity around about 6 to 8 days later. And when the blastocyst develops, it differentiates into two layers. It differentiates into a trophoblast layer and an inner cell mass layer. That trophoblast layer begins to secrete a hormone known as human chorionic gonadotrophin. Now, the human chorionic gonadotrophin is important because it actually maintains that corpus luteum, and this actually maintains this endometrial lining. So what the secretory phase is doing, it's really getting the endometrium ready for implantation. If we don't get fertilization, we don't get a trophoblast layer forming, we don't get HCG, we don't actually have anything to maintain that corpus luteum. And so that actually begins to degenerate. The corpus luteum degenerates. We then lose our progesterone and estrogen. This causes vasoconstriction of our straight arterioles, which then causes ischemia within the stratum functionalis. We lose our blood supply, and that stratum functionalis begins to shed, and then we're back at the start of the menses phase of the menstrual cycle. So if we sort of coincide these regions with the ovarian cycle, remember we have the sort of phases of the ovarian cycle. We have the follicular phase. If I could spell and talk at the same time, that would be really good. So we have the follicular phase, we have ovulation, and then we have the luteal phase. So these are the ovarian cycle phases. So follicular phase is day 1 to day 14, or day 13 I suppose. Day 14 is ovulation, and day 15 to day 28 is the luteal phase of the ovarian cycle. So it's the hormones produced during the follicular phase that drive either menses or the proliferative phase, particularly the estrogen as those follicles are developing, and then ovulation occurs on day 14. And then the secretory phase is driven by the hormones secreted by the corpus luteum. Stimulate my follicle asks, is it the straight arterioles that constrict and then cause ischemia of the spiral arterioles, or do the spiral arterioles also constrict? I think it's probably a mix of both. So it's actually sort of the terminal ends of the straight arterioles that undergo constriction, therefore, and the spiral arterioles, which is causing the ischemia. So the remnant of the straight arterioles remains behind after menses, so we actually still have the majority of the straight arterioles, and then from those straight arterioles the spiral arterioles regrow during the proliferative phase and then further develop in the secretory phase. So I'm not 100% sure about exactly at what level, but generally speaking it's the spiral arterioles which undergo the constriction and the ischemia, and the stratum functionalis is shed. So it may be sort of at the upper level of the straight arterioles. All right, so that's really the menstrual cycle, very messy menstrual cycle here. You're welcome. So we might actually have a look at some of those sections. And so I've got a section of the menses uterus, I've got the proliferative uterus, and we've got the secretory uterus. And so we can actually have a look at what they look like under the microscope. So this is menses here. So here we have the endometrium. Here's the myometrium. You can see some large blood vessels here, and this is all smooth muscle. You can see it's relatively indistinct in terms of the layers. It's not like a nice circular middle longitudinal or anything like that. It's very sort of oblique circular and longitudinal muscle layers. So if we sort of move up here, so here we have down the bottom, we have the stratum basale, and then the stratum functionalis is out here. You can see there's no nice epithelial lining. We can see lots of blood cells. We can see that the glands have become very disrupted here. We can see lots of white blood cell infiltration, but predominantly we can see that we have a very disrupted looking epithelium and stromal layer near the surface here. So you can see how we've got these big gaps with the glands. That's very indicative of ischemia and breakdown of this tissue as it's being shed during menstruation here. So looking for the stroma being shed, blood cells, very unhealthy looking glands. So that's what you look for. Hello, I'm back. When you're actually looking at the endometrium. So again, we've got our endometrium here, we've got our myometrium, and out here we have, let's have a look. Okay, I can't really see a squamous epithelial lining, but generally that'll be the perimetrium on the outside there. Right, so we're going to go have a look at the glands. Here we are. So we'll look at the epithelial surface first of all. So we can actually see a nice epithelium on the surface here. It's ciliated. So the endometrium actually has a ciliated surface epithelium, either described as simple columnar or pseudostratified columnar. Really, that classification's not that important. So this is all the stromal tissue. So if we have a look, it's extremely cellular, and we often say that the stroma of the endometrium is very similar to mesenchyme. So this kind of idea of a primitive connective tissue where we've got lots of cells, a very watery type of ground substance, and not a lot of fibers. And if you sort of think about it, it makes sense because if it's shed, every month, there's not a lot of time for this stroma to actually begin to secrete a lot of fibers and thick connective tissue. So it's a very cellular type of connective tissue. And then we have our glands. So we can actually see our glands here. Well, just zoom out a little bit. You can see most of them are just simple straight tubular glands. You can zoom in here. So here's the lumen of our glands. Here's the base of our glands. So again, while we do have a little bit of space, and that's largely artifactual, it's generally a lot healthier looking than that previous section that we were looking at with the Menses phase. So it's quite a deep type of endometrium. So we're regrowing from our base of our glands in our stratum basale. The stratum basale tends to be a little bit denser with its stroma. And we might be able to see some blood vessels in here as well. If we sort of zoom along a little bit, they're not always that easy to see. We can actually see some blood cells here, but I don't really see a lot of evidence of some arterioles that I can sort of point out, unfortunately. But generally speaking, the majority of the glands are actually simple, straight tubular glands, so cutting cross-section, round appearance, and you can see these straight lines here, very typical. You get a little bit of branching happening down the bottom here, and of course if we think about it, it's actually a continuum, isn't it? So from menses to proliferative to eventually becoming secretory, those glands are going to change over time according to the hormonal profile. Again, here we are in looking at, that was the proliferative phase, yeah, Ruby. So these are the simple, straight tubular glands of the proliferative phase. Nice straight glands, you can see them nice and round in cross-section. If we have a look at the secretory phase, they look very, very different. This is the secretory phase. See how they have this sort of sawtooth appearance or corkscrew type of appearance, and we can actually see material in the lumen now. You can actually see that there. If we go back, the lumen's very difficult to see, and there's very little at all inside that lumen. It's not actively secreting into these glands at this point in time. But in the secretory phase, large, wide lumen, we can actually see material within the lumen there, so it's actually actively secreting, and we've got this very unusual sort of sawtooth type of appearance to these glands. What is the material in the lumen? It's whatever the glands are secreting. So it's generally, there's a lot of glycogen there providing nutrition, but there's other factors which are being secreted which are contributing to nourishing this implanting embryo should implantation begin to take place. we can see some of these spiral arterioles out here as well, so you can see them cut in multiple profiles here. You're welcome. So we can actually see some of these spiral arterioles. They're making their way to the surface and ready to begin to form part of the maternal component of the placenta here. So again, we've got some of these blood vessels here. You can see there's another one here as well. So yeah, if you sort of zoom along, you can often see these spiral arterioles a lot easier. Oh, look at these. How nice are these? So again, multiple sections of that same arteriole, indicating that it's actually spiraling its way towards the surface there. So for my students, my CAM 202 students, you would need to, you can see those three sort of phases, Menses phase, proliferative phase, secretory phase, and you should be able to distinguish between them, put them in chronological order, so Menses at first, proliferative at second, secretory at third, and then answer questions about which hormones are the predominant hormone in those stages and so on. Those are the sort of typical type of exam questions that I ask. I'm not sure what other universities do and what other questions that may be asked. You can see all of the smooth muscle out here in the myometrium. You can see, again, not in nice bands, but just relatively indistinct layers there, and of course lots of vascularization. Okay, are there any questions about that, which we've been talking about? Of course, if pregnancy takes place, then, which one was the last one? No, not that one. Then this is actually maintained. So the corpus luteum is maintained by this human chorionic gonadotrophin. The blood vessels are maintained, the glands continue to be maintained, and implantation continues. All right. So then the next part of the uterus is a little bit different, and that's the cervix. Now the cervix is really interesting. It goes through a number of changes during the lifespan of the person with the cervix. And it's very much driven on the hormones being produced by the ovarian cycle. So if we have a look here, this is sort of an overview picture of the cervix, and we can actually sort of let me just grab my pen here. So now the cervix doesn't have the same sort of structure that the uterus has, so it doesn't really have a myometrium, and we don't really describe as having an endometrium, and it's not actually shed during menses. So it's a very different structure histologically to the rest of the uterus, and it's sort of it's really the bouncer of the upper reproductive tract, I suppose. It really regulates the movement of sperm, for example, depending upon the phase of the menstrual cycle. So, to put it into context here, so the cervical canal would be here, and we can sort of divide the cervix into sort of a junction here from here. So this region here is known as the endocervix, and then from here we've actually got the oops, it helps again if I could spell. So that we have the ectocervix. So the endocervix is lined by a simple columnar epithelium, and the ectocervix that eventually becomes continuous with the vagina is lined by a stratified squamous non-keratinized epithelium. So we actually have this region then where we actually have what's known as a squamo-columnar junction. we get this transition from this simple columnar epithelial lining to a stratified squamous epithelial lining, which we'll have a look at in a little while. Now, during development, the cervix epithelial lining sort of changes. And you might see these little invaginations here. These are actually the endocervical glands. So these invaginations here, these are the endocervical glands. And you can see they're like little cuts inside, or little docking stations for sperm. And interestingly, sperm do dock into these endocervical glands if the stage of the menstrual cycle is actually at the right stage. Now, all of this here, this is actually connective tissue. This is very dense connective tissue. Remember that the core of the uterus, the rest of the uterus, was actually myometrium, which was smooth muscle. Here, this is actually dense connective tissue. And it was sort of described to me when I did my undergraduate that if you feel the cartilage of your nose, that's the sort of structure of the cervix. In a woman who's not actually actively giving birth. So it's actually a very dense, strong, cartilaginous almost density to this connective tissue here. Now, during development, at puberty, what happens is, and I'm not sure exactly about the reason why this happens, and I'm not sure if we do know why, but the endocervix actually grows out into, remembering that the external os, which would be around about here, the external os, the opening. Actually, this endocervix grows out towards the external os, so it sort of projects out into the vagina. Now, at the same time, what happens is the vagina begins, through the hormonal secretions of puberty, begins to secrete glycogen, and glycogen actually begins to feed a native lactobacillus flora that exists within the vagina. Now, the lactobacillus, which exists here, begin to convert the glycogen, and then as a consequence, they release lactic acid as a byproduct. And that lactic acid then begins to drop the pH within the vagina. Now, acid, of course, can be potentially damaging to simple epithelium. We see that in the stomach. So we actually have lots of mucus secretion to sort of protect the stomach from acid. But here we don't have that sort of protective lining. And so what happens is, as the ectocervix has grown out, that ectocervix then transforms back into a stratified squamous, So what I was saying was the endocervix grows out, including the endocervical glands, and then what happens is, with that interaction with the acid, that endocervix reconverts back into an ectocervix, and we get this stratified squamous epithelial lining. Now, you might notice these weird little holes here. These are actually former endocervical glands that were part of that outward growth of that endocervix. As the endocervix grew down, those endocervical glands grew down as well. Then when we get this transformation back into this ectocervix type of epithelium, the stratified squamous non-keratinized epithelium, we actually get a blocking off of those glands ducts onto the surface here. And so these are former endocervical glands that have now become sort of trapped. And so they still undergo the same sort of normal hormonal changes, but now they can actually release their secretions onto the surface here. So these are actually known as cervical cysts or cervical follicles. For some reason, because I'm on the eraser. Or also, sometimes they're known as Nabothian follicles. So what they represent are old endocervical glands. that were existing here, and then through transformation by being exposed to the acidity of the vagina, transform back into stratified squamous, and they lose their connection. So this region here, where you can actually see these endocervical glands and certainly the epithelium, this is often referred to as the transformation zone. And so the transformation zone is actually a clinically important area that when cervical samples are taken, like for example in a pap smear, they actually are trying to sample the cells here to look for any abnormal changes to this lining. How do the lactobacilli get there? I think they're at sort of normal sort of levels normally, but then they actually increased in number through getting a lot more nutrition being supplied by the vaginal cells that now, under the hormonal changes that are taking place, actively begin to secrete glycogen, and so they're providing more nutrition to the lactobacilli. They increase in number, they're getting lots more food, and therefore they're actually beginning to cause the acidity that we associate with the vagina. Does someone who has not gone through puberty still have an ectocervix? Yeah, they still have an ectocervix, and the ectocervix is always there. Same, it's just that they haven't had this episode that happens during puberty where the endocervix grows out into the external os and then gets that transformation happening. Histologically, it's really interesting. So if you see these follicles or these cysts, these are actually natural. This is part of that process where the endocervix grew out, gets transformed through the acidity, and then those glands get blocked off. Okay, so this section, I might just clear that now, close that. So this is the stratified squamous epithelium here. So you can see a very thick epithelium. We've got nuclei all the way to the surface. But what you might also notice is that these cells are very pale staining. So because these are cells are very similar to the epithelial lining of the vagina, they actually also secrete glycogen. And glycogen is an inert substance, and therefore doesn't stain with hematoxylin and eosin. So if we continue our look along, so this is still ectocervix. You can see blood vessels down here. We'll continue our look. I might just decrease here. All right, so we're beginning to get some transformation. We've actually got one of these big cysts here, and it's got those typical sort of endocervical appearance. These are mucus secreting cells which line the endocervical glands. Now, these are part of what, as I mentioned, were a former endocervical gland, and now has lost their connection. You can see where the duct would have been, and it's actually been disrupted, and they can't actually secrete onto the surface anymore. So therefore the secretion continues to build up here and causes these cysts to develop. So there's another one here. So here we're actually starting to get this sort of transformation from this stratified squamous epithelium, and then here we've actually got this simple columnar epithelium. So this is the transformation or the squamo-columnar junction from the stratified squamous non-keratinized of the ectocervix to the simple ciliated columnar of the endocervix. So we can see those cells here, and we can actually begin to see some of these glands, these endocervical glands. You can see them here. They're very characteristic in having this very pale staining cytoplasm because, of course, these are all mucus secreting. Mucus has lots of sugars on the outside, therefore it doesn't interact very well with hematoxylin and eosin. Therefore, these cells are quite pale staining. And so the cervix actually produces a mucus which changes depending upon the stage of the menstrual cycle that the person with the cervix is undergoing. So in a in the presence of estrogen only, the cervical mucus is very thin and watery, a little bit like egg white, so it's quite a non-viscous type of secretion that sort of promotes the motility of sperm through the cervix. Whenever ovulation takes place and we get corpus luteum, and we get progesterone in the system, that cervix changes in structure very dramatically. It becomes very thick. It sort of blocks off the os, the cervical canal, so that sperm can't actually reach the upper reaches of the female reproductive tract, or the reproductive tract of people who identified as female. So again, we've got some more of these glands up here, but again, this is very dense connective tissue. It almost looks like smooth muscle here, but if we go to a trichrome stain section, so where collagen is blue, we can actually see that it's all just dense connective tissue. So again, this is the same sort of section. We've just stained with toluidine blue instead of H&E. Here we've got some of those Nabothian cysts or Nabothian follicles, or cervical cysts or follicles. So we've got our junction here. So this is our transformation zone. This will be part of the external os. So this is the ectocervix, and then eventually that would become continuous with the vagina. So the vagina would be out here. So if we have a look at all this dense connective tissue. Big, thick bundles of collagen running in many different directions, giving it a very solid structure. So again, we've got our stratified squamous, non-keratinized epithelium, pale staining apical cells here, lots of glycogen, so the poor staining. All of this is mucus within this Nabothian cyst. Do the secretions rupture out of the cysts, or is it reabsorb? I think there's probably a mix of both. Sometimes they may rupture. I haven't read a lot about the pathologies that may be associated with the Nabothian cysts, but you can see it is certainly increasing in storage over time. I mean, this one's quite large. You know, if we see a normal sort of endocervical gland here, it's very expanded and relatively large, but I don't know if necessarily that would cause any problems. So here's our junction between our stratified squamous, non-keratinized, and our simple columnar epithelium here, and you can see some cilia associated with this. Again, if we continue on, we can see some of these endocervical glands here. So again, very pale staining. These are mucus secreting. And women, of course, may be interested in their changes in their cervical mucus if they're interested in their fertility. So they may test their endocervical mucus, the cervical mucus, to determine, wow, doesn't that look a little, I'm sorry, I'm a bit distracted there. Doesn't that look a little bit like a Van Gogh painting? All those swirls and everything. Anyway, sorry, I was a bit distracted. So they can actually test their cervical mucus. So there's a test called the spin bucket test where you get cervical mucus on your fingers. If you can stretch your fingers and the mucus doesn't break, it forms these long strings, then that indicates a very fertile period because there's lots of estrogen, no evidence of progesterone, so ovulation hasn't taken place. So it's probably a good time if you want to get pregnant to actually have intercourse. If the mucus breaks immediately, it kind of indicates that there's progesterone in the system, ovulation's already taken place, we've got a corpus luteum present, it's too late to actually get pregnant. So I suppose in a sense, if you don't want to get pregnant, then that's a good indicator that that's probably okay. Again, please use contraception. Don't use my lectures as an indication about whether or not you should be having intercourse. I take no responsibility whatsoever if you happen to get pregnant. To each their own. Please, anyway, let's move on. How about? All right. Any questions about that? I do love the cervix. It's an amazing anatomical structure. Histologically, it's just gorgeous. And then the changes that happen are really quite fascinating to me. I think it's one of the more interesting areas of the human body. Of course, it undergoes quite a dramatic change during birth. We get this hormone release called relaxin, and relaxin causes this connective tissue to actually soften quite dramatically. And so if you remember, I talked about the structure of the normal cervix being like the nasal cartilage. If you feel your bottom lip, that's the consistency of the cervix during parturition when there's relaxin in the system. So it undergoes quite a dramatic change. And then that sort of makes sense because, of course, the cervix, the endocervical canal, needs to expand quite considerably to allow the baby's head through. Progesterone equals thick mucus produced by the cervix. That's correct, Ruby. Yeah, so if there's progesterone in the system, the mucus becomes thick and gluggy. It sort of blocks up that cervical canal and generally doesn't allow sperm into the upper reaches. So some of the ways in which these hormonal contraceptives work is that it sort of prevents ovulation taking place, but two, it kind of tricks the person's body into thinking they're in the luteal phase of the menstrual cycle, and therefore the cervix produces thick, gluggy mucus, and it sort of prevents sperm from reaching those upper reaches of the reproductive tract. So it sort of has a dual kind of effect. One, it sort of prevents FSH release and capture of those developing follicles and ovulation taking place. And two, by preventing sperm from reaching the upper reaches of the reproductive tract. We can allow that question.
Yeah. Oh, sorry. Yep. Why? Yeah.
So Taranese asked, why does thinner mucus facilitate sperm motility? It's just less of a barrier for sperm to actually swim against. So sperm actually swim better in a mucus than they do in water because it actually gives them purchase, something to swim against. But having a relatively thin mucus is much easier to swim in than a much thicker, gluggier mucus. It's sort of a bit like respiratory mucus, I suppose, in a sense that it sort of about trapping rather than actually facilitating movement. So a thinner mucus allows for a better viscosity for swimming in for sperm. All right. Are there any other questions before we move on to have a look at the vagina? I did add some terms into the chat before we started on question or terminology that may be used that may be filtered by the chatbot there, and obviously sperm is not considered an acceptable term, and I missed that one. So James has just talked about the difference in swimming for sperm there. Interestingly enough, sperm swim really fast. So it's suggested it takes about 15 minutes for a sperm after deposition to actually reach the cervix. Then it takes another 15 minutes to actually swim through the uterine cervix, and then another 10 to 15 minutes for them to reach the uterine tube to potentially meet an oocyte. So an hour or so after intercourse, you could potentially contain a fertilized oocyte within your uterine tube, should you have a uterine tube. So it's quite an amazing feat for those sperm to actually swim through. All right, so if there's no further questions, we might move on now to the histology of the vagina. Now, the vagina consists of three histological layers as well. It consists of a mucosa, a muscularis, and an adventitia. Now, the mucosa consists of a stratified squamous non-keratinized epithelium, just like the ectocervix we were just looking at. And you can see how pale these surface cells are. So you can see nuclei all the way to the top. So stratified squamous, you can see they're flattened cells, and non-keratinized, so we get nuclei all the way to the surface, and then very pale cells because there's lots and lots of glycogen being secreted, which actually, as we mentioned, provides nutrition for native lactobacillus, which creates an acidic environment. Now that acidity is really important to prevent non-native bacteria from coming in and infiltrating the vagina. If you think about it, the vagina is moist, it's warm, it's a really good environment to actually grow bacteria and yeasts and things like that. But by having this natural acidity, it actually helps to prevent that microbial growth that we don't want, the non-native or pathogenic microbial growth. So the acidity of the vagina is around about 3.8 to 4.5, something like that, which is around about equivalent to the pH of a tomato, interestingly enough. And I found an interesting blog a few years ago which suggested that your vagina is a tomato in terms of the pH, and anything which may affect the pH may then affect microbial growth in the area. The vagina is an amazing structure. It self-cleans, it self-regulates. Women generally, people with vaginas generally don't need to do much to do it other than just normal showers and so on. As I say, it self-cleans. It has its own bacteria which looks after itself. And as I say, anything which may interfere with that pH then may attract pathogenic bacteria infiltrating. So then we have a lamina propria. So we've got our stratified squamous non-keratinized epithelium. We've got our lamina propria, which contains lots of elastic fibers to allow stretch and so on. And then we have smooth muscle in a relatively indistinct inner circular outer longitudinal layer. Very difficult to distinguish between the two layers. It's not the nice sort of bands which we saw in the gastrointestinal tract, and in the past you can sort of students have asked, how do you distinguish histologically between a section of the vagina and a section of the esophagus? Because they both have a stratified squamous non-keratinized epithelium, they both have an inner circular outer longitudinal layer of tunica muscularis. The difference being there's no muscularis mucosa in the vagina. There's also no glands in the vagina directly. Whereas the esophagus can have submucosal glands in the upper third and the distal third near the junction with the stomach. There's no glands directly in the vagina. Any secretions that help lubricate the vagina come from the cervix or glands near the opening, the greater and lesser vestibular glands near the vestibule there. So there's actually no secretory glands associated with the structure of the vagina. It doesn't have a submucosa. It immediately goes from a mucosa to a muscularis. So we don't have that. And as I say, the tunica muscularis, which is present, isn't in those nice continuous bands like we saw in the esophagus with an inner circular outer longitudinal. It's more of an arrangement of pockets of smooth muscle. You can see actually some of the parasympathetic ganglia here. These are actually neurons, soma of the parasympathetic ganglion here in the wall of the vagina. Interestingly, some larger blood vessels over here, some nerves, and so on. Now I've got a resin section here, so the resolution of this section is really lovely. We can actually have a look at the surface here and see this change in structure from the base of our stratified squamous epithelium to our surface epithelium. And you can see the accumulation of the glycogen in this section. It's really, really lovely. So here these cells are quite acidophilic. We can actually see lots of desmosomes. So it looks a little bit like the stratum spinosum of the skin, doesn't it? This looks like, all those spines present there. And of course, those desmosomes are there to hold the cells together because, of course, the vagina has a potential for abrasion. And so as these cells migrate, can you see these very pale regions around the nucleus, for example? These are accumulations of the glycogen, and the glycogen doesn't stain with hematoxin and eosin. And then as these cells migrate closer to the surface, they become more and more squamous. So the nuclei, you can see, becomes bit more flattened, and we see this increase in glycogen within the cytoplasm, such that the surface cells are these flattened cells with very little evidence of cytoplasmic stains. And we get this nice little layered appearance here where the cell membranes are sort of focused here through the staining. But again, we've got this nice transformation of these round nuclei to these flattened squamous nuclei here. It's such a beautiful section, I think. And really shows that change in structure of this epithelium, the accumulation of glycogen over time. So here's our lamina propria, loose connective tissue. We can see some blood vessels down here. Then as we move down, we can begin to see some smooth muscle cells. Can you see how the smooth muscle cells aren't in one continuous band, but they're actually rather in these little pockets? So here's a little pockets of smooth muscle cells with their centrally located nuclei here. So here's a lovely little smooth muscle cell. But you can see they're in pockets rather than those nice bands. And then here's our outer longitudinal layer. You can see the elongated smooth muscle cells, and they're really lovely. This section is so beautiful. I could spend hours and hours looking at this and find something new. We've got lovely sections of the blood vessels here, so we can see those, some nice endothelial cells. Anyway, as I say, I could spend hours looking at this section, find something new and something amazing to take pictures of. All right, does anyone have any questions about the vagina before we move on? I think we're going to finish off by having a look at the mammary glands, at the different stages of the mammary glands. I do know that we are due to actually finish at this point in time, so we will make this really, really quick. I apologise for us running late. So the mammary glands, again, we talk about them as part of the reproductive system because, of course, the changes that happen within the mammary glands are brought about by those reproductive hormones, because, of course, the mammary glands are there to develop and provide nutrition to the developing baby. Now, initially, that development is driven by a number of hormones, progesterone and estrogen, which, of course, increase during puberty, and we've also get a number of other hormones like prolactin and placental lactogen, which causes further stimulation should pregnancy occur. So the mammary glands consist of ducts and secretory components, and then varying amounts of connective tissue in the forms of collagen and, of course, adipose tissue. So you can see this is a section of the non-active mammary gland. So over here we've got the skin of the areola. The areola, of course, is one of those areas in the human body that is thin and hairless. And we actually have our sebaceous glands actually emptying directly onto the surface rather than via a hair follicle. So it's actually interesting in that respect in terms of integumentary system. So you can see lots of adipose tissue here. We can actually see over here we've actually got some of the duct system. And generally speaking, it's considered that the duct system of the mammary glands is its development is driven by estrogen, and it's the secretory portion which is driven by progesterone. And so here we've got some more of the duct portions, lots of connective tissue, and you can see blood vessels out here, but these are the duct portions. The secretory portions aren't very well developed in an inactive mammary gland, in a person who actually isn't pregnant or anything like that. So we've just largely got this duct structure, which are known as lactiferous ducts here. All right, so and again, this is another section of an inactive mammary gland. You can see it's largely just connective tissue and then just some of these duct portions, these lactiferous ducts. Really not much else going on there. But when the person gets pregnant, then we begin to get these quite dramatic changes. So you can see here now we've actually got these little lobules forming within the structure of the mammary gland. There's a very large dilated lactiferous duct here. So we zoom in here. This is an active mammary gland, but not one which is actively secreting. So this is of a pregnant animal. And so you can see now that the secretory portions are actually quite expanded. And we've got this connective tissue isolating the secretory portions into lobules, a little bit like a normal salivary gland, for example. And then we've got these larger ducts which are carrying the secretions. So these large lactiferous ducts. So this is an active mammary gland, like a pregnant mammary gland. And then when they're actively lactating, it has an extraordinarily different appearance once again. Most of that connective tissue disappears and it just becomes these large secretory components. And you can actually see from this section why it's considered an alveolar type of gland, because it looks like lung. It looks like the alveoli of lung, with the difference being that there's actually milk inside the alveoli. So unless you're inhaling milk, your lung shouldn't look like this. And of course these cells are secretory cells. On the outside we have myoepithelial cells. Those myoepithelial cells are important and responsive to oxytocin. When we get a release of oxytocin, it causes contraction. The milk moves through the duct system. You can see the duct system here, and then eventually out through the nipple. And this is the nipple here. So you can see these large lactiferous ducts draining here. Again, we've got these nice sebaceous glands emptying directly onto the surface here. So you can see just how dramatically the structure of the mammary gland changes depending upon whether it's inactive, active, or actually actively lactating. So this is a lactating mammary gland of, I think it was a cat. So you can actually see all the milk inside the alveoli here. Now, it may not be easy to see, but one of the other features which we may find associated with the connective tissue are, of course, plasma cells, because the plasma cells actually release antibodies into the milk, which helps to provide an early immune system, or a boost to the early immune system of the baby before the baby's immune system is developed enough to produce its own antibodies. So sometimes you can actually find some of those plasma cells out there, and they're usually characterized by being very basophilic in staining. So let's have a look at this one. Yeah. So can you see these very basophilic staining cells? These are actually the plasma cells, and they're actually producing the immunoglobulins, which are being passed on to that early milk. All right.
So Jamie, you may not know the answer to this, but the colostrum, the first thing, the first sort of secretion, which is
colostrum,
colostrum, is very different. Sorry, colostrum's in the brain.
Yep.
Colostrum. Yep.
Yep. I think certainly the components that make up the milk is very different. I think, and I could be wrong here, I think the protein. Yeah, look, I need to go and revise that, and maybe I can bring that to tomorrow's stream. But my understanding is that the levels of protein are much higher in that initial colostrum, and particularly lots of immunoglobulins, very high levels of immunoglobulins, providing that baby with help with its immune system, and then it changes over time. Because of course milk's made up of protein and lipids normally, and so I think it changes over time.
And I certainly know that because the gastrointestinal tract is more permeable to proteins in a baby, that they're more likely in those first few days to directly absorb those immunoglobulins into the bloodstream. Right?
Yeah, so hopefully everyone heard that. So James was saying that the baby's gastrointestinal system is much more readily able to absorb proteins than it is fats in the initial stages, and so maybe that's why the colostrum is higher in protein as well. So Ruby's asked what stimulates the release of milk again by the lactating mammary gland. So the stimulation for milk production is through the hormone prolactin. Now during pregnancy that is in high levels, but in fact high levels of progesterone and estrogen actually inhibit prolactin ability to actually release the milk. So what happens is when the person who's having the baby gives birth, they give birth to the baby, and then they actually, a little while later, give birth to placenta. Now, remembering that the placenta is the site of origin of these high levels of progesterone and estrogen. With delivering the placenta, you now lose that high levels of estrogen and progesterone, you lose that inhibition of prolactin, and therefore the mammary glands can begin to actively release the milk. Now, the actual milk release is brought about by oxytocin. Now, oxytocin is released at the posterior pituitary. It stimulates the myoepithelial cells to contract and move milk through the duct system. It's stimulated by physical stimulation of the areola and the nipple, and it's actually required for maintaining prolactin levels. It is, because it's released by the hypothalamus, which is part of the brain, it's actually a hormonal stimulus, and in fact can be driven by Tarani, thank you very much for the follow. It can be driven by behavioral responses. So a woman becomes a lactating person can become conditioned to knowing that when they hear a baby cry, it's time to actually feed, and then that can stimulate the letdown reflex, which is actually release of oxytocin, causing myoepithelial contraction. And so when a woman hears a baby crying, it can actually activate that letdown reflex, the release of oxytocin, at inappropriate times. So they may not actually want to be lactating at that point in time, and it can bring about that letdown reflex. So James is our neuroanatomist, so he can probably tell you a little bit more about the hormonal control, the nervous system control over that as well. All right, well, I do notice that we were 10 minutes over and we were at least 10 minutes behind. So I do apologise again for our late start. Luckily, we got here in the end. Hopefully you found it useful. So we are doing another stream tomorrow to look at the reproductive tract of people assigned male at birth. So we'll have a look at the testis and all the duct systems, the accessory sex glands, the prostate, the seminal vesicles, and so on, and we can talk about the structure of the penis and erection and ejaculation and all those fun things that may get us booted off Twitch for breaching terms of service. Yeah. So anyway, if there are no further questions, I'd just like to say thank you very much for coming along. I apologise for how messy everything was and me closing the drawing and all that sort of stuff, but we got there in the end. Hopefully you found some of it useful. Anyway, we'll see you in tomorrow's stream. Really appreciate everyone coming along, and I hope you have a good night and see you in tomorrow. Hooroo!